Hello, my name is Gabriel Raulet. I work in the Performance Algorithms and Research Group at Lawrence Berkeley National Lab. I'm here to present our paper titled Distributed Memory Parallel Contig Generation for de novo long-read genome assembly. This work was done in collaboration with Giulia Guidi, Dan Rokhsar, Lenny Oliker, Kathy Yelick, and Aydin Buluç. De novo genome assembly is a problem whose goal is the reconstruction of genomic sequences. So all living organisms have a genome, which is composed of one or more chromosomes, and each chromosome is a sequence of nucleotides ordered in a specific biologically significant manner. And obtaining these sequences is increasingly used and increasingly important for advancing biological research and engineering. The current state-of-the-art method for reconstructing a reference genome involves something called whole genome shotgun sequencing, which is a process where a sequencing machine is given some fragments of DNA from a genome, and the machine reads random subsequences from those DNA fragments, and those are called reads. And de novo assembly specifically is the computational problem whose input is a shotgun sequencing read data set, and the output is a reconstruction of as much of the original genome that the reads came from as possible without any reference. So we have to basically place all the reads where we think they came from and assemble in that order. So to show an example of what that means, here's the overlap layout consensus paradigm, which is commonly used for longer read data sets. And this picture here represents a reference genome. The different colors represent different different subsequences. So these red regions are represent repetitive regions of the genome, which commonly occur, especially in mammalian genomes. So if we align the reads to the original ground truth locations where they were sequenced from by our machine, then we'd obtain a layout that looks something like this. And you can see the machine sequences at a certain depth. This is about a depth of 5 of the genome, and the reads reveal certain facts, like certain sequencing errors that are shown in these yellow rectangles, but also specific biological variants that appear throughout the reads that align to that location. So if we form a string graph by taking sequencing reads as vertices and the overlapping segments as edges, then we can start to move towards getting a good layout of our reads and generating as much of the original sequence as possible. The edges basically represent prefix to suffix overlap. So, for example, this reads suffix aligns within a very high threshold of similarity to the prefix of this read. So in the string graph, we would represent that with an edge. But the string graph in particular has only those edges which are non-redundant and not covered by other pairwise read alignments or overlaps. So if our data set was perfectly if the string graph was a perfect representation of the input, then we get something like this, where we have a linear chain whose root vertices can begin the traversal as we collect sequences along the way. And hopefully the goal is to reconstruct the entire genomic sequence. However, in a real genome, there are repetitive regions, and DNA sequencing machines will inevitably have sequencing errors. So it's very common, in fact it's always true, that you will get these bubble artifacts and other artifacts like tips, and these will create certain vertices that have branch degrees higher than 2, branch degrees of 3 or greater. And this string graph representation still enables us a method of collecting and assembling the unambiguous segments of the genome, the segments that are not that are resolvable. So, for example, here we define the contigs to be the linear components of a graph, which are maximal subgraphs of vertices which are degree 2, meaning vertices that have two adjacent neighbors. So in this example, we would be able to unambiguously reconstruct these three regions, as well as the red region, which is covered by a few reads. So this gives an example of what a real overlap graph and string graph look like. This shows an E. coli data set and the overlap graph representation. And the paper, we had a previous paper here which describes how to transitively reduce an overlap graph into a string graph in distributed memory, and that's listed here. And you can see here our string graph is now ready to be parsed in terms of have its contigs be generated. So our software is called ELBA, which stands for Extreme Scale Long Read Berkeley Assembler. And these are the phases of ELBA. We start with an overlap detection phase where all edges between the reads that need to be there are added to the graph. And then the overlap graph is transitively reduced into the string graph, where we only keep those edges that are strictly necessary. And then finally, the step that we'll cover today is the Contig Generation step, where the string graph is parsed into a set of contigs, and those contig sequences are assembled. The workhorse of ELBA is the distributed memory sparse matrix. So sparse matrices are ideal for storing graph adjacency matrices in distributed memory, because real-world graphs in general show show high sparsity, and additionally, graph algorithms can often be implemented using specific linear algebraic subroutines like matrix-matrix multiplications, matrix vector products, and such. And these sorts of linear algebra kernels can be efficiently parallelized and optimized. So this provides a very good groundwork, or a very good base for ELBA's Contig Generation step. So specifically, we use COMBLAS, which is a library that was developed by Aydin Buluç, and COMBLAS stores large sparse matrices in distributed memory and specializes in performing graph computations on those distributed matrices. So, how do we generate contigs from the sparse matrix representation? On the top here, this is a string graph which has just gone through transitive reduction, and on the bottom left we have the corresponding sparse matrix representation, and I've colored the sort of sub-matrices that a certain local processor owns. So this is a particular processor. It's responsible for storing those edges in memory. This processor is responsible for storing these edges, and so on. So that's how the edges are distributed throughout our processor space. So the first step to finding contigs is getting rid of these branching vertices, which cause these ambiguous paths. So we can identify them by doing a reduction, where we calculate vertex degrees by summing the number of entries that exist in a row and returning that in a distributed dense vector as such. And the branch vertices will be identified as those entries in this vector which are 3 or greater, and the root vertices are those that have degree 1. And we can immediately, once we have this, parse or at least find the rows and columns that correspond to these branch vertices and prune them by simply setting all of those entries to 0, and that the corresponding effect in the graph is that all edges adjacent to those branch vertices are removed from the graph. And at this point, we now have a situation where all of the connected components that have 2 or more vertices are going to be our contig sets. So we have a connected components algorithm that's also already been implemented in COMBLAS for sparse matrices, and we run that, and we are returned a distributed vector which assigns vertex degrees to contig IDs. So to recap, the connected components of the prune string graph are the Contig sets, and each Contig set has all the information necessary to reconstruct that unambiguous sequence. However, there's a particular problem specific to distributed memory, which is that we can't immediately start traversing these contigs because they're not in shared memory. If we just started doing a graph traversal, then basically each time we would go from one vertice to its neighbor, we'd have a high probability that we would need to do some sort of MPI communication. So in order to deal with this problem, we've implemented a load balancing and communication phase that occurs before the Contig sequence assembly, and this helps lower the amount of communication involved and makes our algorithm run very fast. The first step to doing that is the load balancing step. And in the load balancing step, we make the assumption that the size of the contigs is proportional to how long it'll take to reconstruct that contig. So, for example, we assume that contig 0 will take about twice as long to assemble as contig 2, and that gives us a function that maps a contig to the amount of work we estimate it'll take. And so the goal is to compute a partitioning of these contigs such that we minimize the difference in work assigned between processors. So it turns out that this problem is very closely implemented, or it's very related to multi-way number partitioning, which is a optimization problem that is NP hard, but it has a very accurate approximation algorithm that's simple, which involves looping through the contigs from largest to smallest and assigning each contig to the currently least assigned or least amount of work assigned processor. So this approximation takes within about 4/3 of the optimal solution, where p is the number of processors. And I'll describe quickly exactly how that algorithm works. So if we had 3 processors, for example, and 5 contigs, we would assign the largest contig to the smallest bin, which is P0 right now, because they're all 0. We just assign it to the lowest one. We assign C1 to P1, C2 to P2, and then C3 goes to P2 because P2 at that moment only had 3 vertices assigned, whereas the other 2 had 6. And now everyone has 6, so we again break the tie by going to P0. And this gives a very, or more or less well-balanced assignment of work to each processor. So now that we have load balancing, we have to figure out how we're going to actually communicate the information that is needed for each processor to reconstruct its contigs. So the nucleotide sequences themselves are going to be required, as well as the memory associated with the vertices and edges connecting those reads. So all such information is collected together in something called an induced subgraph, and we can form an induced subgraph on each processor according to its load balancing assignment. So each process will be communicated its own local sparse matrix induced subgraph of contigs that it is supposed to reconstruct. And this doing so will basically turn this problem into an embarrassingly parallel problem, where each process simply has to assemble as quickly as possible the contigs that it now has the information for in its shared memory. So to discuss a little bit of the intricacies of what is required to do this, we have to talk about our processor grid. So we were given a distributed vector of assignments from vertices to processors, and in this example we assume there are 16 vertices and also 16 processors. So each vertex would be distributed like so. Each vertex entry in this vector would be distributed along the processor grid in this manner. And if you remember, these processor grids represent which non-zeros are stored on that process if this whole thing was superimposed on top of the sparse matrix of the graph that stores everything. So we want the induced subgraphs, which are defined as subgraphs where the vertex set on each processor is given by its distributed vector assignment from this vector v, and the edge set is given by all the edges that have sources and destination, source vertices and destination vertices within that vertex set. And so we have to communicate each process has to communicate the edges that it stores. It has to send those to its destination based on this distributed vector. However, there's going to be a problem, which is that a given processor doesn't necessarily know where it needs to send its particular edges, because it doesn't necessarily have access to all of the vector assignments, because this is a distributed vector. So there's a very simple naive solution, which would be to do an MPI all gather across the entire world communicator and share everything with every processor. However, this doesn't scale because we will have to do a ton of communication that's unnecessary. So the solution we come up with is to use this processor grid in a more intelligent, a more efficient way, which is to basically observe that a certain given processor, pi sub j, pij, will only communicate non-zeros that it owns locally, which are defined by these relations. So there's a certain slice of edges whose source vertices are in this particular slice, and whose destination are in this particular slice, where p is the total number of processors. So if we instead of sharing everything, if we just communicate the potential source vertice assignments across the rows of every row world, every row in our grid has its own MPI communicator, which we call the row world. And if we do that, we now have access on a given processor to all of the potential source vertices that are in edges that we have to communicate. And if we do the MPI send receive across the transpose, we get the destination, the potential destination assignments of those edges. So we now know the assignments for any given UV pair. We know where we know if a certain edge has a certain U assignment and a certain V assignment that are the same, then we know we can communicate that edge to its destination, because that's what an induced subgraph is. So I'll quickly now describe the assembly process, and this occurs after we've communicated everything, and everything is now locally stored. So each processor has its own local sparse matrix, and in this case we've shown it in a compressed sparse column format, and in this example there's only one contig, but there could be many contigs assigned to a given processor, and the traversal process can now just be done directly using the sparse matrix compressed format, and the particular non-zero coordinate values that depict that tell you where certain reads align to other reads. So we'd start by picking the root vertice, one of the root vertices. There's two of them, which would be a row that has only one entry, and we would collect the non-zero information of where that alignment ends and begins compared to its overlapped edge or its overlapped read, and we would traverse the graph from root until we reach another root, collecting along the way the given subsequences of each read, forming a given contig. And we'll do this for each connected component contig on a given local processor. So we tested ELBA on 3 PacBio datasets, 2 hi-fi datasets, rice and C. elegans, and human CLR dataset, and our results showed very good assembly rates. We can see we only misassembled 2 out of 6,000 rice contigs and 5 out of 4,000 C. elegans contigs, and the completeness score was also very competitive with state-of-the-art software Hi-Fi SM, HiCanoe. Completeness measures the percentage of the reference genome that's covered by a correctly assembled contig. The Contig sizes remain not competitive currently, partly because, or mostly because, we do not implement a polishing or scaffolding phase yet, but we are working on that, and we have seen quite a bit of progress, particularly in prokaryotes with our E. coli data sets. The contigs are almost as large as the Hi5SM contigs. This shows how the speed up rates, which of course, because Hi5SM and Hi-Canoe are not distributed memory, they can't scale up to high node counts. And you can see on rice in particular, which is a larger data set, we can achieve up to 159 times speed up against Hi-Canoe. These are the strong scaling results. They show this particular breakdown. The extraction of contigs is communication bound, so it doesn't necessarily scale particularly well, but it's also very fast in general. So the majority of time is spent doing alignments, and this is true with most assemblers. This is the log plot format of those strong scaling results. And we also ran Homo sapien, a human data set, to show that this scales on much higher node counts as well. To conclude, ELBA is a long-read de novo genome assembler. It's the first software to use distributed sparse matrices to represent graphs, which gives it a much better organization of computation, parallelism, and flexibility in general. ELBA is able to run on a much larger number of nodes than Hi5SM and Hi-Canoe. ELBA load balances contig assembly using greedy multi-way number partitioning, and divides work up using an induced subgraph, a function that we developed. ELBA implements the entire OLC pipeline, and future work is currently being done to resolve repeat issues as well as tips that are caused by sequencing errors to generate much more polished contigs that are competitive with other state-of-the-art software. You can find ELBA here on GitHub. Thank you very much, and I hope you enjoy the conference.